Welcome back guys. Now let's continue with the topic of acid-base balance. Tell me, what is our normal pH? Our normal pH is in the range of 7.35 to 7.45. The acid-base balance or the pH you can say is in the range of 7.35 to 7.45, the value. See, first of all, why do we have to maintain this pH? Okay, because any value less than 7.35 will cause acidosis, and values greater than 7.45 will cause alkalosis. So why it is bad, this acidosis and alkalosis, why is it bad? Because our enzymes are designed in such a way they will only work in this particular pH. Okay, our enzymes, they will work only at this particular pH. If there is any change in the pH, that is either acidosis, alkalosis, the protein structure will undergo denaturation. When the protein structure is getting affected, enzymes are nothing but proteins, right? So enzymes will stop functioning. So any change in pH will cause the denaturation of the proteins that affect the shape of the proteins. So ultimately the enzymes will stop functioning. Enzymes are also proteins, don't forget. Now tell me, pH is important or not for us? It's very much important to maintain, to keep the enzymes functioning, to maintain the homeostasis in the body. So there are three systems, first line, second line, third line. There are three lines of regulation, that is buffer system, lung system and kidneys. These three systems will try to keep the pH in normal range, always in the range of 7.35 to 7.45. So it's that much important to keep the enzymes keep on functioning, to maintain the homeostasis in the body. We have to maintain that pH balance. Now what if the pH changes? You already know. If pH changes, proteins undergoes denaturation. Enzymes, which are nothing but proteins, enzymes will stop functioning. So without enzymes, there is no concept of biochemistry, no reactions, no glycogenolysis, no gluconeogenesis, no protein synthesis, no Krebs cycle, no electron transport chain, no ATP production, nothing. Okay. So pH is very much important for us. First, if there is any change in pH, these buffers will act within seconds. Okay, if there is any change in pH, the buffers will act within seconds and tries to bring the pH back to normal. Okay. So three lines of regulation, buffers, lungs and kidneys are there. Now first of all, my question to you is, God created this pH 7.35 to 7.45. First of all, why is it changing? Why is it changing in the first place? Why our pH is fluctuating or trying to change? Because always in our body there is production of acids. Okay, because of the metabolism, always we produce the acids, guys. Now these acids releases H plus ions. Okay, H plus ions can decrease our pH, that is acidosis. So constantly every second in our body, because of metabolism, constantly H plus ions are getting generated, acids are getting generated. Okay, so there is a chance that you can end up in acidosis. So is that good for you? Definitely not. So that's why there is a buffer system in our body. So whenever this H plus is getting generated, every second in our body this H plus are getting generated. Whenever this H plus is getting generated, we have to neutralize it. So for that neutralization, there is a buffer called as bicarbonate. So this bicarbonate is nothing but base. H plus is acid. So acid will react with base, or you can say the base is nothing but the buffer. Now H plus will react with HCO3 minus, becoming H2CO3. So becoming H2CO3. Now this H2CO3 is going to split into water and carbon dioxide. So finally what happened? We have neutralized this H plus. We have neutralized this H plus with the help of this bicarbonate. So bicarbonate is a buffer which will neutralize acids. So who produces these bicarbonates? Our GIT produce bicarbonates, kidneys produce bicarbonates. So to neutralize this acid, we are having this buffer. Done. Next step. This bicarbonate is considered as the most common buffer in our body. And where is it present? In the plasma. So the most important extracellular buffer, because it's present in the plasma, it is neutralizing the H plus that is getting produced. Any other buffers are there? See, other buffers include hemoglobin. Okay, hemoglobin which is present in the cell, that is RBC. So hemoglobin also acts like a buffer. So it's a intracellular buffer. How? Let me tell you. Guys, look at here. This is your RBC, let's say. What is this cell? RBC. Now, your tissues produces carbon dioxide. We all know our tissues produces carbon dioxide, the CO2 production. Where the CO2 will go? CO2, thousands and thousands of molecules of CO2 is going into the RBC. Now, in the RBC, this carbon dioxide reacts with water, forming H2CO3. Now, this H2CO3 with the help of enzyme carbonic anhydrase is going to split into HCO3 minus 1 bicarbonate and H plus. So, we have produced one HCO3 minus, and where this HCO3 minus will go? The HCO3 minus will go into the plasma, and that's fine. Okay, good. HCO3 minus is going to the plasma and it's going to act as a buffer over there. But the problem is here, we have generated one H plus, one proton. H plus is generated, one H plus and one HCO3 minus. Now, thousands and thousands of molecules of carbon dioxide will produce thousands of molecules of this acids in the RBC. So, tell me, is it good or bad to have this acid in the RBC? Definitely bad. So, in order to neutralize this H plus, there is a protein called as hemoglobin. So, now look at here guys, this red color protein which I am showing you, see, let's say this is the hemoglobin. Hemoglobin is made up of what? Amino acids. One such amino acid is histidine. So, here I am showing you what is this amino acid in the pink color. This is the histidine amino acid. What this histidine amino acid will do? The histidine amino acid will accept the H plus, will bind with the H plus. So, neutralizing the H plus. So, the H plus which is in bound form, the H plus which is bound with the protein will not contribute to any H plus. Only this free H plus, the free H plus will contribute to pH change. But once the H plus is attached with the protein, it cannot contribute to any pH change. So, now this hemoglobin, by accepting the H plus, acting like a intracellular buffer. Because of which amino acid it is acting like a buffer? Because of histidine amino acid. So, now tell me, it's the amino acid responsible for the buffer action is histidine. Hemoglobin is a intracellular buffer in the RBCs. Next, not only that, in our urine also. Tell me, in our urine, intercalated cells releases the H plus. So, we don't want the H plus to be alone because the urine will become acidic. Do you want to produce too much acidic urine? No. So, in the urine, there are molecules called as phosphates and ammonia. This phosphate and ammonia, where they are present? First of all, they are present in the urine and they act as a buffer. So, tell me, who is the buffer in the plasma? Bicarbonate. Who is the buffer within the cells? Hemoglobin. And who is the buffer in the urine? In the urine also we have buffers, that is phosphate and ammonia. If there is any free H plus in the urine, they will go and bind with the H plus, neutralize the H plus. Done. So, with this, what we have completed? We have completed the buffer topic. Buffer acts within seconds trying to correct the pH back to normal. Done. Now, what is the role of lungs in the maintenance of pH? Does lungs can change your pH? Yes. Now, try to understand. Look at here guys, there is a lady. Now, what she is doing? She is crying. Now, when a woman cries, it's not just crying, it's with lot of emotions, lot of drama included. How she is crying? She is crying with hyperventilation. She is crying like this, okay? She is doing the hyperventilation. Okay, you know how a woman cries. Now, tell me, whenever she is crying, let's imagine she is doing hyperventilation, guys. Hyperventilation. Okay. Now, tell me, whenever she is doing hyperventilation, something like that, she is removing more carbon dioxide out of the body. So, there is increased carbon dioxide washout. Okay, there is increased carbon dioxide washout. More carbon dioxide is going out of the body. Now, carbon dioxide, I have told you, it's a source of acid. Carbon dioxide is a source of acid in our body. When you are removing the carbon dioxide, what happened to the acid production? The acid production in the body decreases. Okay, carbon dioxide is a source of acid. Carbon dioxide reacts with water, produces H2CO3. Later H2CO3 split into H plus. So, normally remember, carbon dioxide is acid. Carbon dioxide is acid. Now, when a person is trying to do the hyperventilation, the person is taking the carbon dioxide out. When the person is taking the carbon dioxide out, what will happen to the acid production? Decreases. So, when acid production decreases, that will lead to respiratory, because of respiration, right? Respiratory alkalosis. Okay. Now, this female can land up in respiratory alkalosis. So, what I am trying to say, see, the way you breathe can change your pH. The lungs can actually change your pH. Simple. If you do hyperventilation for 1-2 minutes, if you try it, look, don't do it. But if you do hyperventilation for 1-2 minutes, you may develop respiratory alkalosis because you are taking the carbon dioxide out of the body. So, when you are taking the carbon dioxide out, the acid production decreases, that can lead to respiratory alkalosis. Okay, fine. See, for just understanding purpose, here I am showing you a crying female who is doing the hyperventilation. But in reality, tell me who will do the hyperventilation? The person who is having hypoxia, the person who is suffering with hypoxia. Who can suffer with hypoxia? The person who is in high altitudes, the person who is living in high altitudes, or the person who goes to high altitudes. Okay, the person is climbing the hill, now he is in the high altitudes. Whenever he is in the high altitudes, he will develop hypoxia. Now, whenever there is hypoxia, we all know the person will start to do the hyperventilation. Now, tell me, whenever the person is doing the hyperventilation, the person will go into respiratory alkalosis. Means the way you breathe can actually change your pH. Now, exactly opposite. Let's say the person is doing hypoventilation. Now, opposite, that is hypoventilation. Or the person is having respiratory depression. So, he is doing hypoventilation. Whenever you do hypoventilation, what will happen to the carbon dioxide levels in your body? The carbon dioxide level build up. There is more carbon dioxide. There is more carbon dioxide retention. Okay, there is more carbon dioxide retention in the body. Now, carbon dioxide is going to increase the acid production. Carbon dioxide will lead to more acid production. Now, whenever there is more acid, there will be respiratory acidosis. So, at the end of the day, the point which I am trying to teach you here, the way you breathe, either hyperventilation or hypoventilation, can actually change your pH, can actually lead to acidosis or alkalosis. So, your lungs has a role in the changing of pH. How this is helpful for us? Let me tell you. Let's say the patient is suffering with acidosis, guys. Because of some reason he is having acidosis. Now, tell me, is it good to have acidosis? No. Immediately, do you know what our lungs will do? Our lungs will start to do hyperventilation. Why? To remove the carbon dioxide out of the body. So, immediately the person will do hyperventilation to decrease the PCO2, to decrease the carbon dioxide. Whenever carbon dioxide decreases, what will happen? Acid production decreases and pH will start to increase. pH will start to increase. Okay, coming back to normal. So, first the patient is having what? The patient is having acidosis, right? Acidosis means what? Low pH, which is bad. So, you have to bring the pH back to normal. You have to increase back to normal. So, lungs can help you in that. So, lungs either by doing hyperventilation or hypoventilation can actually changes your pH. If you do hyperventilation, you can get into respiratory alkalosis. If you do hypoventilation, you can get into respiratory acidosis. So, lungs can actually change your pH. Okay, done. Now, what is the important MCQ for your exam? The person who is living in high altitudes. Okay, the person who went to the high altitudes, he will suffer with hypoxia. So, he will have mountain sickness, right? Have you heard about mountain sickness? So, tell me, in a person who is having mountain sickness, what kind of abnormality can be seen? So, because of the hypoxia, the person will do hyperventilation that can lead to respiratory alkalosis. So, yes, mountain sickness, the problem is respiratory alkalosis. The pH is increasing, respiratory alkalosis. Okay, remember that. Next, let's discuss what is the role of kidneys in the pH balance. How kidneys will help in pH balance? See, kidneys, that is the third line of regulation. Third line means it takes days to correct pH. Takes days to correct pH. Kidneys takes days. Buffers acts within seconds. Respiration minutes to hours. But kidneys takes days to correct the pH. Okay, what happens if the patient is having acidosis? Tell me, acidosis means more acids. Now, in these conditions, do you know what kidneys will do? Kidneys will increase the bicarbonate reabsorption. Okay, kidneys, what they are doing? They are increasing more bicarbonate reabsorption because we want the bicarbonates in this condition to neutralize the acid. So, to neutralize this acid, what do we require? We require more base reabsorption. Kidneys will increase bicarbonate production. Okay, and the same kidneys will increase acid excretion. Increase acid excretion. Okay, so these are the three things which I want you to know. Whenever there is acidosis, what kidneys will do? Increase the bicarbonate production, increase the bicarbonate reabsorption, and increase acid excretion. We don't want acids, right? In the condition of acidosis, we don't want acids. So, we have to kick the acids out. So, how these three things are happening? If you see these three things, the topic will be completed. Okay. Now let's begin our topic with how bicarbonates are reabsorbed. We all know 80% of the bicarbonates are reabsorbed in the PCT. How? Let me explain you. So let's take the heading bicarbonate reabsorption in PCT. Okay, where bicarbonates are getting reabsorbed in the PCT. How, sir? Look, this is the lumen side, means this is the place where urine is going. Okay, here is the place where urine is going like this. Now in the urine, what is the most important molecule that is present? It's the bicarbonate. Bicarbonate is freely filtered. So now tell me, you have to lose this bicarbonate? No, we should not lose this bicarbonate. They are the bases, they are buffers. We have to reabsorb it. How we can reabsorb this base? See, these bicarbonates we have to reabsorb. So in order to catch a fish, you have to put a bait. In the same way, if you want to reabsorb this bicarbonate, see, you are keeping a bait. You are putting a bait, that is H plus. So with the help of a transporter, see, there is a transporter. The name of this transporter is called as proton, that's a H plus H plus sodium exchanger. Okay, sodium proton exchanger. See, the sodium is coming into the cell, proton is coming out. So this is called as a sodium proton exchanger. Now tell me, who is secreted? H plus is secreted. We all know H plus attracts minus, negativity attracts minus, negative charge, positive charge is going to bind. Now this HCO3 bind with the H plus becoming H2, two molecules, right? H2CO3. Now what we have generated? H2CO3, carbonic acid. Now this carbonic acid, with the help of enzyme called as carbonic anhydrase type 4, with the help of carbonic anhydrase type 4, this H2CO3 is going to split into water and carbon dioxide. Okay. So what we have produced ultimately? Water molecule, carbon dioxide. We all know water can cross the cell membrane, carbon dioxide can cross the cell membrane. So now the same water and carbon dioxide is going into the cell, crosses the cell membrane, going into the cell. So now right here, water coming into the cell, carbon dioxide also comes into the cell. Now reverse reaction is going to happen with the help of carbonic anhydrase type 2. So with the help of carbonic anhydrase type 2, again reverse is going to happen. Now this water reacts with water reacts with carbon dioxide, forming again H2CO3. So again H2CO3 is generated. Okay, now this H2CO3 again going to split into one H plus, okay, and HCO3 minus. So finally topic completed. Now we have reabsorbed this bicarbonate, right? The bicarbonate which was supposed to be in the lumen, finally where it came? It came into the cell. Okay, this is not newly generated. So the bicarbonate reabsorbed indirectly. It's not just the direct reabsorption, because these are negative charges, right? Negative charges you cannot take directly. So indirectly you have reabsorbed. So finally the bicarbonate is reabsorbed. You have got your bicarbonate. Now where this bicarbonate is going? We want the bicarbonate in the blood, not in the PCT cell. Okay, so now this bicarbonate, with the help of a transporter called as sodium bicarbonate cotransporter, with the help of the sodium bicarbonate cotransporter, bicarbonate goes into the plasma, and here it acts like a buffer. Okay, here it is acting like a buffer. So this bicarbonate is going to act like a buffer. So with this, what we have completed, guys? We have completed how bicarbonates are reabsorbed in the PCT. See, what are the important things that you should know for your exam point of view? What if this sodium bicarbonate cotransporter is mutated? Okay, right, sodium bicarbonate cotransporter mutation. Once the sodium bicarbonate cotransporter, if it is mutated, do you think you can reabsorb this bicarbonate? No, you are not going to get this bicarbonate. Now without bicarbonate, no base means acidosis. So the person can go into the acidosis, metabolic acidosis. Without bicarbonate, metabolic acidosis. So the mutation of the sodium bicarbonate cotransporter will lead to a disease called as renal tubular acidosis. Okay, renal tubular acidosis type 2. So now from today you will be getting a question. Okay, from today if you ever get a question something like this, renal tubular acidosis is a defect in which part of the nephron? It's a PCT defect because of the mutation of sodium bicarbonate cotransporter. Okay, there is loss of bases, you are not getting this bicarbonate, there is a defect in the bicarbonate reabsorption. So always remember, RTA type 2 is a PCT defect. It's a PCT defect, that is bicarbonate loss. You are not getting the bicarbonates are gone. Next, what else you should know? Pharma. in pharma point of view, so there is a drug called as acetazolamide. Now what this acetazolamide will do? Acetazolamide is a carbonic anhydrase inhibitor. It's a carbonic anhydrase inhibitor. Now the day you take acetazolamide, carbonic anhydrase is not going to work. And when these carbonic anhydrases, when they are not working, do you think you can reabsorb the bicarbonates? No. So the day you take the carbonic anhydrase inhibitor, now this bicarbonate is going to be lost in the urine. So write acetazolamide causes bicarbonate loss. So can lead to acidosis. Can lead to acidosis. So now my question to you, where do you want to lose the bicarbonates? In which condition do you want to lose the bicarbonates? Where do you want to lose the bases, guys? In a patient who is suffering with alkalosis. Who suffers with alkalosis? Okay, who suffers with? Tell me, give me an example where a person is suffering with respiratory alkalosis. Mountain sickness. Have I taught you? So, in a person who is suffering with mountain sickness, we all know there is respiratory alkalosis. So, in a person who is suffering with alkalosis, what we have to do? We have to kick the bases away. We have to kick the bicarbonates out of the body. We don't want the bicarbonates now. We want the acid now. So, the drug of choice for the mountain sickness is, the drug of choice is going to be acetazolamide, which is a carbonic anhydrase inhibitor which causes the bicarbonate loss and it can lead to acidosis. Done? So, finally, can I say something like this? PCT cells secretes acid, true or false? The proximal convoluted tubular cells are secreting acid. Yes. So, proton is secreted from PCT, true. Next, what will happen to the urine pH? Will the urine becomes acidic or basic in the PCT? Students, look, who is getting secreted? Acid is getting secreted. When the acid is getting secreted into the urine will become acidic, right? The urine in the PCT will become acidic? Answer is no, because there is base. So immediately this buffer, bicarbonate is a buffer, no, will neutralize the acid immediately. So urine pH is unchanged. The urine pH in PCT, urine pH in PCT is unchanged. Urine pH in PCT is unchanged because there is bicarbonate buffer, because there is HCO3 minus bicarbonate buffer. So tell me, who is the most important buffer in the PCT? The most important urinary buffer in the PCT. So the most important urinary buffer in the PCT is, yes, bicarbonates are there. These bicarbonates are acting as a buffer. Doesn't allow the urine to become acidic. Done. Next step. Let's see how bicarbonates are generated. See, this first topic completed. Bicarbonate reabsorption in the PCT completed. Now, how these bicarbonates are produced in the time of acidosis? In the time of acidosis, we have to produce a new bicarbonate. Not only reabsorption, we have to produce new bicarbonate. So how that is happening? Look guys, bicarbonate generation, especially where? In DCT. Okay, so it's not PCT, it is a DCT. So in your book, in this material, it is given brush border here. Actually, there is no brush border. It's a mistake. This is not PCT, this is a DCT. Again, I am telling you. Okay, here brush border shouldn't be there, but it's given. Now, how these bicarbonates are generated? There are two methods, guys. There are two methods by which these bicarbonates can be generated. What is the method number one? Simple. Method number one is, we all know cells produce a carbon dioxide. Cells uses oxygen and produce a carbon dioxide. So this is the carbon dioxide. And we all know cells contains the water. In the cell cytoplasm, water is there. So carbon dioxide and water are anyway there in the cell. Now when you mix this carbon dioxide and water, what you can produce? H2CO3. This H2CO3 with the help of enzyme called as carbonic anhydrase type 2. I have told you, in DCT there is only carbonic anhydrase type 2, but PCT there is carbonic anhydrase type 2 as well as type 4 are present. Now, DCT, right? Carbonic anhydrase type 2. With the help of this carbonic anhydrase type 2, this H2CO3 is going to split into one bicarbonate and one H+. So tell me, now you have generated this new bicarbonate, right? See, the bicarbonate, this is the newly generated. See, it is not getting reabsorbed. I am not saying it is getting reabsorbed. It is newly generated. It's a de novo synthesis of this bicarbonate by using carbon dioxide and water, by mixing carbon dioxide and water with the help of carbonic anhydrase type 2. So yes, we gain a new bicarbonate. But during this process, something bad happened. What is something bad? One acid also produced, one H plus also produced. Bad. Now what we have to do? We have to neutralize this H plus. How? How to neutralize this H plus? We are kicking it out of the body. Okay, now we are kicking it out of the body with the help of a transporter called as proton pump. Proton pump or hydrogen ATPase. You can say hydrogen ATPase or proton pump. So with the help of this proton pump, the H plus is coming into the urine, kicked into the urine. Now tell me, do you want to keep this H plus in the urine? Do you want to keep this acid in the urine? No, we don't want it, because it can lead to severe urinary acidification that can lead to the damage to the nephrons. So in the urine there is phosphate HPO. Okay, there is HPO4. Okay, there is HPO4. So in our urine, what is there? There is a buffer. Phosphates are there. Now what these phosphates will do? Now this phosphate will accept. Now the phosphate is going to accept this H plus, becoming H2PO4. Okay, becoming H2PO4. So now tell me, who is acting as a urinary buffer? In the urine also there is a buffer, that is a phosphate. The phosphate will accept the H plus and becomes H2PO4. Okay, so yes, phosphates are considered as urinary buffers. Now, what is the clinical importance here? What if this proton pump is mutated? This proton pump, this is not working. It is mutated. Once it is mutated, do you think acids will go out? No, the acids are not going out. The H plus is not going out. So that will again lead to renal. That will again lead to renal tubular acidosis type 1. So RTA type 1 is due to defect where? Is due to defect in proton pump, is due to defect in DCT. So DCT defect. Okay, DCT defect lead to RTA type 1. PCT defect will lead to RTA type 2. In RTA type 2, there is a sodium bicarbonate cotransporter mutation. In RTA type 1, it is a proton pump mutation. Their bicarbonate absorption defect. Okay, their bicarbonate absorption defect. Here, acid excretion defect. Here, the acids are not getting excreted. There, the bicarbonates are not getting reabsorbed. So, anything, either bicarbonate loss or acid accumulation can both lead to acidosis. So, done. Next, let's look at the second way how we can generate the bicarbonate. In the time of need, whenever there is a need, so for example, whenever there is acidosis, now these DCT cells, this is DCT, no? Now what these DCT cells will do, from the blood they will take this amino acid called as a glutamine. Okay, they will uptake more and more glutamine, which is nothing but amino acid. Now what will happen? Now this glutamine will undergo breakdown. Okay, now the glutamine will undergo breakdown into NH3 minus ion, okay, ammonium ion and 2 bicarbonates. So now see, we have produced 2 new bicarbonates, guys. 2 new bicarbonates are produced and you can reabsorb it. And what these bicarbonates will do here? The bicarbonates can act like a buffer. Okay, the bicarbonates can act like a buffer. But during this process, you have produced this NH3 minus, right? Now where this NH3 minus is going? It is going to the urine. See, NH3 minus ultimately came into the urine. Now urine, what this NH3 minus will do? It will minus NH3 minus. This minus will attract the proton. It will attract the H plus. So becoming NH4, So now finally, can I say this NH3 minus acting like a buffer, sir? It is accepting the H plus. So acting like a buffer. So now tell me, what are the urinary buffers? The 2 important urinary buffers at the level of DCT. The answer will be phosphates and ammonia. Okay, these phosphates and ammonium ion, they are acting like a buffers. So finally, what are the products? H2PO4. So this H2PO4 and NH4. See, they are called as titratable acids because they are also slightly acidic. See, this HPO4, okay, sorry, H2PO4 and NH4, these are slightly acidic substances. Okay, slightly acidic. So because of them, our urine will be acidic. So tell me, why our human urine? Okay, human urine is slightly acidic. True. Why? Because of the presence of this H2PO4 and NH4. So these are titratable acids. These are called as titratable acids. So urine is acidic, slightly acidic because of NH4 and H2PO4. Done. So with this successfully, what we have completed? We have completed how bicarbonates are generated. Tell me in how many ways the bicarbonates can be generated? 2 methods. What are they? By mixing carbon dioxide and water, and by the breakdown of an amino acid called as the glutamine. Glutamine breakdown yields how many bicarbonates? 2 bicarbonates. Okay. Now this proton pump mutation can lead to which type of RTA? RTA type 1. RTA type 2 is due to defect in PCT. Okay, RTA type 1, let me write here, RTA type 1 is a defect in DCT, defect. Next, now let us discuss about the intercalated cells. Sir, what are these intercalated cells doing? Now, these intercalated cells, they are having some mood swings, guys. Imagine you are having a, a girlfriend or wife, she will be having a mood swings, right? Sometimes she behaves differently, sometimes she is more romantic, sometimes she is more like a devil, okay? So, what I am trying to say, according to the situation in the body, like acidosis, alkalosis, these intercalated cells, they will change their function. For example, see, in the time of acidosis, do we need acid? In the time of acidosis, we don't want acid. We have to excrete the acid. So, look, in the time of acidosis, this intercalated cell, what it is excreting? It is excreting the acid into the urine. Acid excretion. Okay, there is acid excretion. But the same intercalated cell, in the time of alkalosis, see, in the time of alkalosis, what it is doing? It is excreting bicarbonates. So, sometimes they are acid excreting and sometimes they are base excreting according to the situation in the body, and that's actually good. During acidosis, we want to excrete the acid. During alkalosis, we want to excrete the bicarbonates. Okay. So, now this acid excreting cells are called as alpha intercalated cells. So, these are alpha intercalated cells, and this base excreting cells are called as beta intercalated cells. So, intercalated cells can be alpha or can be beta. Alpha intercalated cells are acid excreting during acidosis. Beta intercalated cells are base excreting or bicarbonate excreting during alkalosis. Okay. So, yes, kidney can sometimes excrete acid, can sometimes excrete bases according to the situation in the body, trying to help in the pH, maintenance of the pH. So, with this, what we have completed, we have seen, okay, what are the different mechanisms by which the kidney can control our pH. So, I have told you, kidney can increase the bicarbonate reabsorption. Kidneys can increase the bicarbonate production, two ways of bicarbonate production, and kidneys can increase the acid excretion. During acidosis, kidneys excrete acid, but the alkalosis, during alkalosis, kidneys excrete the bases. So, by these three different mechanisms, kidneys can actually regulate our pH. So, with this, what we have completed, the kidney's role in the maintenance of pH topic completed. Hope the video is helpful. Thank you.